through. Dude. I don't know. I feel like
What's our catchphrase today?
Toyotathon.
No, we can't do catchphrases again. We already did it last time.
What else can we do?
I'm not the director. I'm just the camera guy.
What?
All right. Our cameraman said he's not our production executive anymore, so we need some guidelines and some tips on.
You got TikTok light?
We used Whistle Up as a catch
Sure.
in our last video.
Mm-hmm.
He doesn't want us to do a ticker anymore, so we're going to need some things to say or do or jokes or something to keep these kids interested.
Oh.
So.
Yeah.
Oh.
I think.
Oh. Okay, we're back, guys. We're back, Honors Bio, and today we got some baby spinach. You might be asking, what are we doing with baby spinach? Well, we're going to study photosynthesis. So, Mr. Kujula, Mr. Yvette, we're going to take you through this lab that involves the process we're going to learn called photosynthesis. So, let's go ahead and take a look at all this stuff. Let's just walk through some of the things we've got here that we're going to need.
So at the end here, we got a hole punch. All right, that's going to be used to cut the discs out of the leaf. Next one here, we got some syringes. They do have caps on the end, so we'll have to take those off. But otherwise, a little syringe there that we'll put the leaf discs in, as well as the solution. Then we got some stopwatches here, just to keep track of time. We do have different color of cellophane as well, so that we can look at how different wavelengths of light ultimately affect the rate of photosynthesis.
Here we got sodium bicarbonate, okay, which is a fancy term for baking soda, and we're going to learn that this is going to give the plant CO2 in this water. So when we start talking about sodium bicarbonate, all we're really saying is we're giving the plant CO2. Okay, we got two solutions, and they are different. The one in the plastic beaker is one that has this in it, okay? So it's the one that has the CO2 in the water. And then here we've got just plain water, okay? So you might want to hypothesize what's going to happen with these two different solutions. This one is going to have CO2, this one's not. And what does that mean for photosynthesis? Okay? Let's get into it. All right, so first thing we got to do, Bulldogs, is we got to punch some discs. So you avoid the vein and you just take some, boom, there you go. Okay, we're going to get a total of around 10 of these puppies, and then we grab some forceps and we're going to put them in the syringe. So we had to pull the plunger out, boom, boom, and we put them in there. All right, Bulldogs, so in this syringe here, we got these leaf discs, spinach leaf discs floating right now, and this is in the water solution, so it's going to be our negative control. And we'll have time to talk about what that means a little bit later. And Mr. Kujula's syringe, he's got the solution with the CO2 in it from the baking soda. But right now, if you take a look, they're still floating. So what we have to do is we have to sink these discs. And the way we're going to do that is we're going to use the pressure of the syringe to push into the spaces of the leaf the solution that's in here. And once that happens, these leaf discs are going to become dense and they're going to sink to the bottom, more dense. Okay, so let's do that. So you swirl, push, swirl, pull.
Now, what I like to do is push a lot of the air out, just until you just get a little bubble water coming out. That way there's no air in there whatsoever. Take my cap, and I'm going to go ahead and pull. Little bubbles will form as and then when I release it, I can push that push that solution back into the leaf discs. And you can see them start to fall. And I'll go ahead and swirl it around, and I'll do that a couple more times here. gonna go ahead and empty out some of that water. We're not gonna use that again, so it's okay. That way I can pull a little bit more air out. Give it a good shake. I'm actually gonna push down on the table this time.
Louise. Holy shnikes.
Oh
Yeah, that's
my
gonna
gosh.
happen. It's gonna happen
the
if
heck?
you take your
My
thumb
finger
off of
just
that.
slipped.
Yeah. Uh-huh.
Oh,
All right, I think I got these sunken. They're ready to rock and roll. All right, let's go put them on the lab table and start this up. All right, so we got just a general one here. This is just how it works. Over time, we would count how many discs are floating. Every minute, we'll count how many are at the top. Okay, so we got our timer. You're gonna need a timer for each one. This one's kind of going fast. A normal situation. They're producing oxygen bubbles as a product of photosynthesis, and that's what's making them buoyant and float to the top. Okay, so this is kind of a normal situation. Okay, now we're going to show you guys some independent variables where you could test to see if we could speed this up or slow it down.
all right. So what we have here are all of our independent variables set up and our control. In the red cellophane bag, we have our independent variable that's going to test the red wavelength of light. It's going to filter out every wavelength of light, excuse me, except for red. Next to that, we have our extra sodium bicarbonate. So this is going to be a little bit more sodium bicarbonate, a little bit more CO2 present for those leaf discs in the second syringe here. The next one is going to be our regular control. So there's just our normal amount of sodium bicarbonate. And then here we are going to be testing the light intensity. So we scooted it a little bit closer to the lamp so that we can see that added light intensity and how that affects the rate of photosynthesis. To help us check our rate of photosynthesis, we have a timer here. So we'll go ahead and start the lamp and start the timer. And we'll wait to see which leaf discs float.
There we go. Bingo.
Bango.